So today we're going to talk about some basic steps on single cell RNA-seq data analysis. I will start from importing the count matrix using a 10x cell ranger output and show you how you're going to QC the data and how you do some filtering to remove some empty barcode and noise, low expression genes, and then normalization. Then we're going to use dimension reduction techniques to visualize the data. The major step for single cell data analysis is cell type classification. So we're going to spend a lot of time to talk about what the techniques in Partek flow to help you identify your cell type. And then after the cell type classification, all the downstream analysis like a differential analysis, biological interpretation is very much the same as other RNA-seq like bulk RNA-seq data analysis. All right, I'm gonna turn off the. Okay, so today the data we're gonna get is from 10x cell ranger output. I think from usually if somebody already run the cell ranger for you, they will give you the cell ranger output folder. So those are the files containing in the output folder. What we need is there's a filtered H5 file, and also there's a filtered feature barcode matrix folder, which containing 3 files. And typically, if you download this file, and that's what they put on the website, but if you have the file, the name might be different, but they are in a certain folder called filtered feature BC matrix.h5. And you can append whatever the sample name for you. So this is a one format. And the other one is a folder. It's also called a filtered matrix barcode folder, and it containing 3 files. Either they might be zipped or unzipped will be like this. One is a matrix, MTX, and then barcode.tsv and gene.tsv. So 3 files per sample. And this has exactly the same information as the filtered feature barcode matrix H5 file, but this file will be much bigger than combine all those 3. I would highly recommend you get this H5 file, but we also support to import this 3 file format. Okay, so I'm going to use the H5 file to start building the analysis pipeline. All right, so once you log on to Flow, as you can see, this is a new interface. If you ever used Partek Flow before, maybe you can see it's a little bit different. You transfer file to the folder in your account, and then the next step is you're gonna generate, get a new project. Also, if you want to find help, under help menu, we have how-to videos, we have the documentation. If you click on that, and there are some live training recordings over here. We'll always put our training recordings in here. Okay. Okay, so now I'm gonna create a new project. Let me just call that, CRA-seq demo. Give a meaningful name of your project. And now the first step is you're gonna add your data. You click on add data, and we have videos and documentation. If you are very new to Partek Flow, you can find the videos here. So click add data, and on the top, it will choose your technology, whether you're doing single cell level or bulk level or some other stuff. For single cell level, there are different format, and typically, 10x count feature barcode matrix. Those are 3 files. There are some other vendors also use this kind of format, like a Flint Bioscience and a Parse Bioscience. They also have this type of format. And 10x, they also have H5 format. Or if somebody give you a full count matrix, like a BD single cell, they will have a full count matrix. And if somebody already using other tools like a Seurat to process your data, you can directly import a Seurat object, which with the RDS format. Or if they're using Python and give you a H5AD format, or even you can start from FASTQ file to generate the count matrix yourself. And also, we support spatial transcriptome data, ATAC-seq, VDJ, and flow mass spectrometry data. So the wizard, you can use the wizard to import the data. And today I'm going to use H5 file, select this format, click next. You browse to the folder containing the file. And for me, I have it, actually, it's right here. I select my H5 file. If you have multiple samples, you can select them all within the same folder. Okay, and click next. First, I would give a meaningful name. Try to make the name short, so later on, and also informative. So later on, when you plot your samples, you don't want the label take most of the resource, right? So I just call that, 1K ppmc data. And Partek knows how to parse that. In this data, we have 1,200 cells with 33,000 genes. When your upstream analysis generate this count matrix, they will have an annotation file to generate the matrix. And usually they will use the ensemble database. So it's better that you find from the upstream that what annotation version they are using to generate this count matrix, and you're going to match the version. So typically, you have to specify the species assembly and then what ensemble version. But as long as your species version is, human HG19, HG38, or mouse MM10 or MM39, the assembly version is correct. The minor change of the gene annotation, the ensemble version, maybe won't make a whole lot difference. Okay, but it's better you get the exact version, so they will match. So suppose they are using ensemble 108 version to generate that, you specified it. And then within this H5 file, there are two identifiers for each gene. One is the gene name, one is ensemble. Ensemble is a unique name, but gene name is more meaningful to you. However, sometimes some genes you might, have two IDs, but that's very, very few number of genes. And I will just choose this one because it's more meaningful. You won't have, well, if you have a duplicate, suppose there are two gene names here, will automatically deduplicate the genes. So the gene has to be unique, your barcode has to be unique for each sample. And this containing raw count data, but if you import data that somebody already processed the data, you can choose the normalized count option. The key thing is you have to know your data is in linear scale or log scale. This is very critical because for log data, you can have negative value, but for linear scale data, you should not. But this one is raw count. We know your minimum is going to be 0. And we can import all the features, or you can choose during import, I can filter out the genes. If the gene, there's no expression in any of the cells for all my samples, you can do that because, 33,000 genes, probably 20,000, they don't have any expression in any cells. Also during import, you can choose to import only cells if the read count is less than a certain number. If you use a cell ranger filtered count matrix, I think the default, I never see they will include any cells that they have less than 500 UMI count. So they already filtered. Even you apply this filter, it won't filter anything. But if you download the raw count, you might have like 1 million barcode. Probably, you only have 20,000 barcode that really have any read. So it will be helpful to eliminate some barcode they are empty. And how you define empty, you can specify how many UMIs in that. I will just use the default like this. If you want to be very permissive, you can say, okay, if a barcode has even 1 read, I will import that. You can type in 1 here. But I typically use the default and then click finish. So you will see the progress bar. We're importing the data, and once this progress bar finished your task, whenever you see the task is transparent, that means that the analysis or the task is running. You mouse over, you will see the progress bar. But once it's finished, it will become solid color. And the first progress bar will disappear. You're going to see a solid data node. And when you mouse over, you will see, okay, this is one sample, and there are 1,200 cells and 33,000 features and 13 megabytes. Also, if you click on the metadata, you can add more data to your if you want to import more samples, you can add more. Okay. All right, so now if you click on this data node, also you can add data from here too, if you will. If somebody already analyzed the data, you can add all the annotation information, but since this is raw, we don't have anything. And the first step I would like to do is to perform QA/QC. There are different ways to QC the data. You can QC the genes to look at the gene distribution, or you can QC the barcode to look at the cell information, or you can look at, do more filter. Just this is already, you think it's a cells with the filter, and this is the raw barcode. If you have a raw count matrix without filter, you can look at this to look at the knee plot. Or look at the sample level or report. You can explore all the other options at your own space. Because the time limit, I'm gonna just do one. So definitely you want to perform single cell QA/QC. And this task will only generate a report, and this report will be a test report. It's rectangle, it does not generate any output. So when you click on that, you can look at the details of the task, or you can look at the report. Whenever you open the report, you can either double click to open this in the current browser window, or you can right click to open in a different tab of your browser. It depends on your habit. So for this QA/QC report, we see there are 4 plot, 2D scatter plot displayed in what we call the Partek data viewer. Okay, so we're going to use the Partek data viewer to look at the data a lot when you do single cell data analysis. Now let's talk about the data viewer. On the left side, this is the control panel. You can choose to save this session. So you just save the data viewer. I will save one to show you later on, or you can save as a different name. And you can export the image. So the image export will include all those 4 plots. Whatever displayed in the plot area, you can export as a PNG or a PDF or SVG format. Define the how big the size you want. And if you have something, rotating, I will show you later. If you have a 3D plot you're rotating, you can capture a video and put in your PowerPoint. And you can put in the notebook. And then this part set up is showing you, if in the data viewer, how you start from scratch to make a plot. And then there's 2 questions you have to answer. The order doesn't matter. One is what type of the plot you want to make, bar chart, pie chart, a scatter plot, etc. Once you choose what style you want to use, and then you want to say what information I want to display. I want to display based my raw count, normalized count, or whatever. So you answer those 2 questions to make a plot, and we will talk about that as we go along. Once the plot is up there, you can select, select the dot or select the bar, etc. So this is, you can manually select it or use a certain criteria to select. We'll talk about that later as we go along. And for single cell, once you select the cell and you want to classify, and the classify means give a name. Okay, this is a T cell, this is a B cell. So then you will call that a classify. So this is you annotated cells. The bottom here, the configure, is showing you some more advanced option to configure your X, Y axis, and the font size, tick mark, etc. So all this, you want to make a perfect plot to put in your publication. And the style, you can rendering the plot based on look at the color, shape by, etc. All those information we'll talk about more. And a grouping and some descriptions. So different plot as you selected will have different options for the configure. And here I have 4 plots here. Once you click on it, you will see the border will be thicker. That means right now you are selecting this plot. And within the plot, once you mouse over in, you will see within the plot there's more controls. And here, this is a handle for you to drag it. You can drag this, and suppose I want to put it right here. And so you can rearrange your plot and to the way you want, then you can export the image. But if you change your mind, we have undo. We have a lot of undo for you to go back. A lot of redo and undo here. On the right side, you can duplicate the plot. You can download this, export the plot. You only export this particular one, and then send it to a notepad, a notebook. We have another tab called notebook. You can write notes for your project. Also, if you want to focus on this particular plot, you can click on the maximize button and then see some more details. If you scroll your mouse wheel in one direction, you can zoom in and press down your right mouse. You can pan it to go to a different location and zoom in. Scroll your mouse wheel the other direction, you can zoom out. Okay. Let me just, okay. So I can see everything. And then this is a reset button. So you can reset to the default. And then this one will close this plot, will remove from your data viewer. The vertical part, there is handles. We're going to go, this is just a different selection mode, and you can use a pointer to select. You can use a pencil to paint it, or you can draw a bounding box or draw a circle, or you can lasso it using different shape. And once you select some point, suppose I just select some point, and then this one is the invert selection, just invert the other way. And this is just reset the view. And this is to turn off the legend and just to rescale the data or flip your X and Y axis for a certain plot. Okay. All right, now this is the basic component of Partek data viewer, and we can, as we go along, you're gonna see we'll use all those options and all those handles when we try to perform a certain analysis. Okay, now let's get back to the single cell QA/QC. All those scatter plot, each dot is a cell. So the legend, as you can see, there are 1222 cells. And the first plot, the y-axis represents the total UMI count. As you can see, cell ranger, the exclude all the UMIs less than 500. The minimum is 500. And the x-axis is random spread out all the point. So otherwise, some point will be on top of each other. So it's not very meaningful. Also, we put a violin plot, and the width represent the density of the distribution. So we can clearly see, most of the cells, they have less than 20,000 UMI count, right? There are some, they're high, too high, it could be a doublet, and too low definitely is not good cell, right? So this is one perspective. The second one, even the cell have a lot of UMI, 500, but you can see some cells, the y-axis represent number of detected genes. There are only 12 genes detected. Probably, if you, for human, initially the annotation has 33,000 genes, but it only detected 12, probably is not good cell, and you want to filter that. And too high, again, it might not be good either. This one is mitochondrial count. For human and mouse, when you import the data, when you specify annotation, we know which genes is on a mitochondrial chromosome. We use that information to calculate genes on mitochondrial chromosome to divide by the total count. So if you have a cell, mitochondrial percentage is too high, and depends on your study. This is PBMC data. If it's too high mitochondrial genes, usually that means that the cell is dead or is dying. It's not a good cell, right? But if your study like a muscle or something, you might expect high mitochondrial percentage. And some people saying mitochondrial percentage for PBMC shouldn't be greater than 10%, or somebody said even 3%. You really should look at your data and determine your cutoff. Also, for human and mouse, we calculate the ribosome genes and give you the ribosome percentage. Again, too low and too high, still not going to be good. But different study, again, you have a different criteria. So I would highly recommend you, based on those perspectives, to perform the initial filtering. When you filter your cells, statistics basically is a filtering, right? So when you filter cells, always look at the feedback. There's no gold standard cutoff. Any cutoff is gonna be subject to your data. So use your common knowledge based on your biology expertise, what study you're doing, and remove some cells you think they're really not good. And it should be, I think this is the first step you're filtering. Try to be more permissive, because as we go along, we're going to filter more. And after you filter, your data size will be smaller, so your downstream analysis will be much faster. And also, since you remove noise, that is also good for you to find the signal. So when you filter the data, first you have to select them, meet what criteria. You can manually say, hey, I want to select this range of the cells. You just manually select it. So once you select them, and all the other plot will reflect where the points are when you select it. Okay, but this one is kind of too arbitrary, and it's hard for people to replicate, even yourself. Every time you won't be able to exactly select the same thing. So I would highly recommend you to go to the select and filter. Use a rule. You can build a rule to select your cells, and you can manually do that. But if you choose using criteria, and here we display those 4 criteria. That's the range, minimum to maximum. When you mouse over, you can see the histogram and the distribution. Also, if you click on this, you can pin it. So make the histogram always stay there. So now you'll see, oh, okay, that's the range. And we can drag this sliding bar. Okay, I want to make the selection, like here, or you can manually type in a value here. Okay, so it depends on what you want to use. For example, I really don't know what I'm going to filter based on the total UMI count. Maybe I can start from number for detect genes. Detect 13 genes is really too low. Maybe I will say, okay, let me just use, you have to have detect at least 200 genes. And too high, it could be doublets, right? And then here I probably say, okay, let me just use 5,000. Once you press down, press enter, and this bar will be updated. And also this filtering, this selection will be reflected on your plot too. And you can see here, for every plot, there are this many cells selected for all of them. And you can see, those are, and also the deselected point, you can make them the color dim it, or you can make them gray, or at a certain point, you don't want to differentiate that, you can choose, hey, don't change anything. But I'll just use, for this one, I want to see which one is excluded, or which one is selected, which one is not. And in the meantime, for the mitochondrial percentage, if I say, hey, I don't want to include anything greater than 10%, I type that in. Then you look at the data. Oh, half of my cells are gone, and that's too much. Maybe this criteria is not good for this particular cell, particular study. I'm going to change that. So always look at your data, look at the feedback, and to make a decision. And don't just say, okay, well, one paper they use a 3%, the other paper use 20%. What should I use? Always look at your data, and have the common sense to filter it, not too aggressive. If you don't know how to use ribosome percentage to filter the data, then don't use it. Okay. So suppose this is the criteria I'm going to use. And then to select my cells, I think this is pretty good. I still got 1,000 cells there. And then you can save this session. So if I save this session, I just call this, cut off. Now, if I close everything, log off Partek, and later on you go to the data viewer, and go to the select and filter, you can see what you are using here. So we will save the status, what you during when you click the save button. So later on, you'll know, oh, okay, this is my criteria. Okay, always remember to save some important stage as you go along the data analysis. So now this will only select them. I didn't filter, and here is a filter include. This is a filter exclude. So I will choose a filter include, or you can filter at this. You can save at this stage too. It's up to you. But this filter only in this data viewer session is not reflected in your analysis pipeline. So when you save this session, next time you will know the filter is in the data viewer. If you want to apply to the pipeline, you're going to have to apply this filter in the data viewer to your pipeline. So because you filter observation means your cells, your samples, or sometimes you perform filter on feature, that's apply filter feature. Okay, so I'm going to apply this observation filter to my data, to my analysis. Now Partek will ask you, okay, which data nodes you're going to apply the filter? You only have one. There's no other choice. So this guy, I want to select it. And then there's a filter task will be starting, and the Partek will tell you, okay, it's in the queue if the server is busy, and then you can refresh that. And now I can safely get out here, and this is the breadcrumb, and you go to where your pipeline is, because I didn't save the filter session. So now you can see I perform a filter. Before I have a 1222 cells or 33,000 features. Now, if you mouse over this one, you will have a 1053 cells, and still we didn't filter features, right? So your data size got reduced. It's always a good practice to clean up your cells from your data. And the next step I would recommend you is a good practice is to filter genes. So basically, I want to reduce the matrix and reduce the number of rows and number of columns, right? So we only filter one dimension. Now I'm going to filter the other dimension is to filter genes. Also, any circle count matrix, you can perform a filter from here. And if you want to filter cells, you can also build a rule to filter cells. If you say, I want to base on my number of expressed genes is greater than such, and or use this option to perform exactly the same filter. But this one, you don't have the visualization. You just know, okay, I'm gonna use, like if you use a Syrah, you just use this number and to put a name there to give it a try. And what data analysis is iterative. After you filter, even once you get to the very end, you feel like, okay, this result I couldn't find my genes. You can always come back and redo the filter. Okay. So now I'm going to filter features. Feature means genes, means proteins, means ATAC regions. The measurement. So now I'm going to click the filter. There are different options for you to filter. The first one is noise reduction. Basically, we filter out noise Usually we call that low expression. If a gene has only one read count, do you think that's really an expression of one read, or do you think it's a mistake, right? So usually you cannot differentiate the low expression versus noise. We can just remove all the low expression genes. But how low is low expression? This is always a good question. You have to define your own. If the maximum value of the gene expression across all my cells is less than such and such, I will call that low expression. Or if my total gene expression sum up all the cells of this gene is less than such and such, I will call that low expression. But my recommendation is if this gene expression is less than such and such in a percent of the cells. It depends on, everybody has their own strategy. I like to use, try to be more permissive. I will say if the gene expression is 0, you don't have a less than 0, or you can change that to 0, right? It's 0 in 99% of my cells. So you have to do a little calculation. I have 1,000 cells in total, and 9% is 0. That means only 1% is non-zero. 1% means only 10 cells. 10 cells probably is not a population, right? Unless you really study really minor population, you think 10 cells is a population. So again, it always depends on your study and depends on your data. You're going to determine the parameter. So I will use this one for this particular data as a filter. And another option, sometimes if you say, I want to just grab the top, 3,000 genes with the biggest variation or biggest, dispersion, you can choose any statistic, just grab the top genes. And most of the time when you use a Sera in their tutorial, they recommend you to do this way. So it's kind of aggressive for visualization, but you will capture all the major signal. There's always pros and cons. Another one is I want to filter based on the gene annotation. Suppose in the ensemble annotation, they will have a feature type. If you say, I want to filter if the gene biotype is, protein coding, or you can choose whatever to filter it. The first iteration, you probably don't use that. Another one is I can filter based on the list of genes, and we can generate a gene list, or you can manually type in or copy and paste a list of genes, like filter include or exclude. If I know some genes is noise genes, like mitochondrial genes or ribosome genes or cell cycle genes, I want to filter exclude. You can type that in. Typing is, I'll just, only one CD4, just only one row per gene. There's no delimiter. Just do it this way. Okay. All right. So here I'm gonna use the noise reduction to do my initial filter. And it will perform a task here. And as you can see, it will progress. And if you know what you want to do, you don't have to wait until this task is finished. You can click on it and keep on building your analysis. So the next step, and you have to do, is normalize your data. The normalization, everybody has their own opinion, and we have some like a Sera, SE transform, or if you use ScanPy, people use a SCRAN deconvolution, whatever your favorite, but I would recommend you, this is a Partek normalization, recommended. So basically we normalize each cell, make all the cells on the same scale. So we use a CPM and add 1 and then do a log transformation. So after this type of normalization, your gene expression value, your 0 is still going to be 0. And your non-zero value, even the raw data, you have 1 read, you probably have pretty high value, probably it's a 3 or 4. And then your expression probably won't be bigger than 20. So you kind of have a range about your data, between 0 to 20-ish. But also you can, if you really want to know the distribution of the data, you can check the feature distribution. So the important thing in Partek pipeline is different data node containing different information. So this data node containing normalized read count. It's the same number of genes as over here. Okay, now we can see after the filter feature, we got 12,000 genes here. Okay, I think if you have more than 10,000, it's pretty good to do the downstream. But this one containing the raw count. So this is very important because later on, when you try to make a plot, which data node do you want to use to make a certain plot? Okay. All right, so after normalize, now we are ready to do any analysis or visualization. Typically, we want to see the data, and seeing is believing, right? But the data is very high dimension. You have a 12,000 dimensional space for each cell, and the human can only visualize a 3D. So we're going to rely on some dimension reduction technique to map the data to 3D or 2D. The first step you're going to do is a PCA. And PCA, I will just say use the default and click finish. So PCA will grab the major component and grab the most informative dimensions and present it to you. But for PCA has some drawbacks here. It's good to look for the bigger difference, but not a smaller difference. So for single cell data, people want to, based on PCA, they want to, they are all under exploratory analysis. They will look at the t-SNE or UMAP. You probably heard those terms. They are all dimension reduction technique, map high dimension to low dimension. It's not a clustering method. It does not give you p-value. Any judgment you make based on those plot is gonna be subjective. Okay, and also based on PCA, you can do t-SNE also. Today we don't have a time. I'm not gonna do t-SNE anymore, but if I click on the UMAP, this data node containing UMAP score, it does not have a gene expression, and PCA is the same. You only have a PC scores for those 1,000 cells. There's no gene expression. Just remember that. And one way to invoke the plot is you can double click on it to open it, or you can click the task report. If I double click to open, and the default in my configuration is a 3D scatter plot. You can configure to 2D scatter plot, in your private, in your settings. I don't have time to go over that. But this is a 3D scatter plot of UMAP. Just visually, we can see this data, this 1,000 cells. I can visually see there's a cluster over here. There's a cluster over here, right? But what are they? Yes, this is the question you're going to answer for single cell data analysis. That's the major step. So if visually we see some clusters, but I don't really know, do these statistically, based on the gene profile, do they really a cluster? I would recommend you perform cluster analysis using statistics to partition the data. So the first thing you want to do is, based on PCA, you use a graph-based clustering. The nice thing about graph-based clustering, you don't have to tell it how many groups you want to partition the data. It will figure it out, okay? However, the one thing is, so you can change. There's advanced, every time when you see advanced option, that means that there's more tuning up for this. If you don't know what to do, the default is a not too bad option, but it won't be, it's the best option for your data. That's why you have to try it. I would recommend you, pay attention to the resolution parameter. Of course, there's other parameters will also affect your result, but this one, if you mouse over here, it will tell you, if you make this number bigger, increase this number, will have more cluster. Decrease this number, will have less cluster. So if you run your initial using the default settings, so suppose you have too many clusters, and then the next round you say, I want to have a less number of cluster, change the value, to a smaller number. And then if you apply it and then finish it, that will give you a different number of a cluster. So I just want to show you an example that how, if you want to configure the result, okay? So after graph-based cluster, if you double click on it, graph-based will only give you the annotation of your data to say, okay, how many groups are partitioned for each cell, what I will assign a cluster membership. It's only called a cluster 1, 2, 3, 4, A, B, C, D, it doesn't matter. It's not biologically meaningful. So sometimes people say, oh, I can do your analysis, give you the graph-based analysis result. This is definitely not the final result. That's not what you want, right? You want to give the biological meaning. And 1, 2, 3, 4 in Partek just means cluster 1 has the most number of the cells. And then we just rank the different clusters, and the bigger cluster will be on the top. And this is the percent of the cell. So this is what graph-based annotation will give you 4 groups using these settings. And also during I forgot to talk about this. If you click on the rectangle, you can click rerun this. I will open the dialog. By default, we'll also compute biomarkers. This in Partek is using t-test. After you partition the cluster, I have 4 clusters. I will compare each cluster versus the non-this cluster and use a fold change to give you the genes as upregulated. This is all the by default. That's why you see the computer marker, and the result will tell you, cluster 1 compared to non-cluster 1, those are the top genes is upregulated. It's almost tell you those are the positive biomarkers. And whether you agree with it, you have to verify. So this is, as a biologist, your job to do the identification. Maybe you will see some familiar genes, CD3D, CD3E, and this kind of my T cell markers, right? And in this one, I see like a CD14, maybe, related to monocyte. And this one, CD79A is the biomarker for the B cells, 79B. And here, like cytotoxic cell markers, right? So it will point you to the direction, will help you identify your class. So we are gonna use the information visualization statistics only based on the gene expression profile to generate the result, and the biomarker information. Now we're gonna do the cell type classification. Okay, so let me just open the UMap again. And over here, if you say, oh, how I see there are 1, 2, 3, 4, 5, 6 clusters. How graph-based partition the data into 4 clusters? Are they kind of land up with what I see? So you probably want to use the graph-based annotation to overlay this on this plot. So one way is you can use the configure, go to the style, and this is very important. You have, like what I said, each data node containing different information. You have to know which data node containing graph-based annotation, right? It's only in the graph-based, just to show you the pipeline, only this data node containing those 4 cluster information, not here, not here. So you don't have that. So first of all, you have to make sure you choose the correct data node in order to select the annotation. So you can click on this data node. This is a little icon that will show you the pipeline. And you can search or use this triangle, you can expand or collapse all the information to search for this thing. But you can type that in graph-based, type in graph-based, Partek will help you to search. So this data node, and once you mouse over, you see the minimap. When you click on it, that will change, when you select the graph-based annotation, you can see it's highlighted. It's kind of in the darker. So that means now I'm in the data node that is, has graph-based annotation. Now, if I select from this drop-down, that will display in the graph-based annotation what we have. It has a graph-based annotation. So this pop-up will show you all the attributes of the cells. And when you see the blue, that means the categorical information, and the green means the continuous information. And we saw the number of expressed genes, the total count, ribosome, etc. So they are, you can use them to color, but they are in green. They are continuous variable. So now I can use the graph-based annotation. Okay, so this is one way to do. I will show you some other ways later. So now let's take a look at this. Visually, I can see, oh, this red, graph-based is saying this is one cluster. That's pretty good. But some of them, they are not consistent. Like cluster 1, I think is only over here, but actually graph-based is saying, okay, this dot is similar to over here. And this dot is similar not to this green, but to here. So you will, and then the yellow is, I got 2 clusters here, right? So because UMAP, you only see one perspective of your data, and a graph-based is also, human cannot see the full picture of your data. That's why we have to use different perspectives, try to piece all the puzzles together, right? So when they are consistent, it's easy for you to make a decision, but when they are inconsistent, it's going to be hard. You might make a mistake, or you need more information. Okay, so next, even, we see this is the green, the red color is one cluster, but we still don't know what cell type that is. I cannot just call that cluster, 2. I need to know what biologically meaning of this cluster. So now you are the expert in the biology. You have to have some knowledge about your canonical biomarkers. We're going to use your knowledge to identify your cell type. What I like to do is I can add my biomarker table here. So now I will show you how to set up a new plot. I can choose, what information I want to use. I use get data. I will search my biomarker data node, and this is the biomarker data node. Once you mouse over, when you see the background is green right here in my mouse, the area, that means it's draggable. Okay, so this is the minimum. I cannot move my mouse. And you can press it down and drag it. And the blue is called a drop target. You can, drop it right here. I put the bottom of this plot, and for this biomarker, you can only make a table. So that's the only choice. You cannot make anything else. Also, there's videos. Whenever you use a dialogue over here, you can click on this how-to videos to learn the software. Okay. So now I have the biomarker table over here, and I can see, I got, 25. But if you download, there's a download button right here that will give you more genes and will give you p-value and the fold chain, etc. You can use that to make your own cutoff. But the top, by default, will only give you the top 25 genes as the biomarker. I like to rearrange this to be, supposed on the right side. Also, in the meantime, what I like is to duplicate this plot. I put it to the bottom. So those 2 plots, they are exactly the same, but I want to use different information. This one is the graph-based information, and this one I want to look at my biology. I start from the genes I know. CD3D is a T cell marker, right? So when you mouse over, this is a green background, means it's draggable. I can drag it, and now I can put on the axes or I can color it. So now I can clearly see, remember this is logged data, logged expression value. This is normalized count. And if you go to the style, you will notice that this data node right now, we're on the normalized count data node, okay? So the values are from 0 to 11. We can clearly see, this group of the cells, they are have a high expression of a CD3D. But this visually, we see this is one cluster, but you can rotate that to a different location. You probably see, oh, there are two different clusters here, right? So probably you're very sure this is T cells, and you're not very sure, whether this one. And if that's the case, what we can do is one gene probably is not enough. I can use a different genes. You can also choose a different genes to color it. Or if you want to use both genes at the same time, what you can do is when you drop, there are 3 triangles here. That means you can use up to 3 genes to color your data at the same time. So I put the CD3D on the green channel, and then I put the this is a CD3E. I can put a CD3D on the red channel. Okay. Now, when you see, if I zoom in, when you see the red, if you mouse over, you will see the CD3D and CD3E expression value. Click on it, you can see this is a single positive. Only one gene got expressed. But if you look at the yellow, both have a high expression. Okay. Well, remember, single cell is not perfect. Sometimes you have a dropout. Even this gene, this gene is, this cell is really T cell, and the reason the CD3E is 0 is probably you didn't capture it, right? It's because the technology. That's why nowadays everybody tries to do multi-omics. So you want to use a different perspective, try to capture the information. You can use a protein, for example. Anyway, that's how much answer we can get from here. So you can use another genes to color that, or I can use I know the other one, like a CD14 is a monocyte. I want to put a monocyte on the red channel. Oh, I can see monocyte mainly over here, and then this is all green, this is all red. And maybe I know, B cell markers is a CD I put on the blue channel. So you can clearly see, just using one good marker, I can see these separate all the different cluster, right? And the reason I want to have this up here, so I can go side by side. Statistically, this is one cluster, right? Visually, this is one cluster, okay? And this is a B cell marker genes, and maybe this is a B cells, right? And this is a monocyte. But sometimes if there is inconsistency, you have to make a decision. When you color it, you can only use 3 genes to color them at one time. But if you say, I know some marker genes for this cells, and this will be more than 3 genes, what are you going to do? So if that is the case, you're going to build a gene list. You go to your username, click on, go to the settings, and on the left side, there's a list. So click on the list. You can make a new list. You can either browse to a file. It's going to be a text file with only one column. It's a list of a gene name. Or you can copy and paste your gene list here, like a CD3, CD4, CD14 or whatever, just one copy and paste, make a list. And also on Partek website, we host some gene list here. And based on this is the literature, you will say this is not the standard. This is just only from one paper here, okay? Just remember, everybody tries to build a database. You might agree with the database, you might not. So they're going to change all the time. This is what we have. And you go to the different, I'm going to make this a little bit bigger and go to different pages. Right now, we have some biomarkers for the immune cells and for neural cells. And you can, mainly as on human and mouse. So you can check it and add it to the list. That will add to this list. And from each list, if you click on it, you can download the list as a text file, or you can edit. You can only edit the name or description of the list. You cannot edit the content because we try to avoid, if you change it, your previous analysis will be invalid. if you look at this gene list, you say, oh, I don't agree with that. I want to add more genes. What you can do is just download this gene list and then edit it and then upload it, make your own list. Okay, so that's how you make a gene list, feature list. You can use the feature list to filter your data. And now we're going to use that to rendering the data. Okay, so if I want to look at my B cell markers, I have 92 genes here. They are all positive biomarkers. How do I render that? If I use this plot, you have to use a style and make sure you're going to use this gene list based on the normalized read count, right? Use a certain expression to do that. So make sure you choose the normalized count data node, which is already my default here. And then you're going to choose the color by feature list. And you're going to choose your feature list. Suppose I have B cells. And now this is the metric is using the total count of those 92 genes. You add them up for each cell and then color the cell. Or you can use the mean, whatever, the scale will be very similar. It's only the maximum is different, but the color probably will be very much the same here. Okay, I can clearly see using the B cell marker genes, this group of the cells is kind of, B cells, if I agree. So that's how you use the color to rendering this. Suppose based on this color, I want to select some cells. I know this is, whatever the cell type. If you want to make a decision, so for example, I want to show you a couple of ways to select and classify. Based on either plot, it doesn't matter which one. So suppose I want to manually select, if you want to manually, you might want to zoom in to make it a little bit bigger to make your select better. And also if the point is too small or too big, you can go to the style and change the size and make it bigger or make it smaller, whichever you think will fit what your purpose. So suppose I really want to, based on statistics, it said it's one cluster. Based on visually, I think it's one cluster. And based on, earlier I look at my marker genes, I think this is a monocyte. So you want to make a decision here. You can use one of the selection point. This is the paint. You can define the paint size, as you go along, that will select. Okay. I think this is very useful when you do transcriptome, use your histology information. Or you can choose this lasso and you just manually lasso it. Double click to close the lasso. So now it will select. I selected 341 cells. Now if I, reset it, that's how I selected. Make sure once you selected, your legend will always tell you select how many cells. And also it will reflect on the other plot too. Once I select it, you go to classify. This is manual selection. And now you give a name. So you classify selection. I will call this monocyte. So now this is you really classify the cells as monocyte. Okay, so this is manually select, but sometimes, only when you really crystal clear, especially on the visualization, they're very separate from all the other cells. I think this can be useful. Most of the time, you probably don't want to use this. Instead, if you say, hey, graph-based clustering showing a pretty good, I think of the cluster 1 over here. Based on graph-based clustering, I think a cluster 1 is T cell, okay? Instead of manually select them, you go to select and filter and choose the criteria. And in the criteria, of course, you can go to the graph-based data node and select the graph-based annotation. And there's an easier way to do. This legend is draggable. You drag this legend and drop it. This is a blue background. That means the drop target. Drop it right here. It will automatically select the right thing. So I have 4 clusters. And now this time you say, hey, I want to select the cluster 1. So when you select cluster 1, it will include the cells over here and also over here. And if you think, you know, that makes sense, I will use this to, you know, call that, once they're selected, you go to classify, call that T cell. Just classify and give a T cell. But sometimes this is, you know, if you don't like that, I can show you another one. If you say, I want to base on gene expression instead of for graph base, or using this both criteria, what you can do is you can drag a CD3D. Suppose I want to use a CD3D and a CD3E, both of them. If I want to select cells, both gene expression, they have to be high expression. So, and also include, you know, graph base called a cluster 1. Let's see what we have. You can use the sliding bar to, you know, choose your cutoff. I'm gonna just manually, you know, type in. I would say, okay, the maximum is 6, is 10. I will use a 6 as minimum. I will call that high expression. See, 340 cells selected, right? And then on top of that, CD3D has to be high expression. I still use a 6. Now you filter all this. You can have as many genes as you want. Keep adding all the genes. You can, you know, select your normalized data count, normalized count data node, and search for, you know, suppose I want to search for CD8. Add another criteria. I want the CD8 has to be high, you know, greater than 5, whatever this. So you can add as many genes as you want, but this is kind of too much, right? Most of them, they are not. I'm gonna remove this guy. I always look at the feedback and look at the visualization, you know, combine all the information visually, statistically, biologically, you know, all this together, you make a decision. Okay. So suppose this 332 cells, I think they are T cells. They are selected. I classify selection. I will call that a T cell. Okay. Now I got a 2 cell type classified. And then the other one for the B cell, I want to show you another option, because for the B cell, we use a group of genes, like 92 genes. I don't want to specify, you know, add all the 92 genes in my criteria. Instead, I want to use the sum of those 92 genes. So if you go to select and filter and choose the criteria, I'm going to remove all those criteria. And from the drop down, once you color one of your plot using the gene list color, you will have a placeholder called the gene score. And the score right now is the sum, right? For B cells, you can, this is a continuous variable, and click on that, and we can take a look at the histogram. You know, now you make a decision. Okay, what do you want to call that high expression? So suppose I want to use maybe 150. Okay, I select those cells. Let's take a look at all the plot. So over here and some of over here, right? They are selected, and then I will classify. There are 179 cells selected. Classify selection. Now I see a yellow warning. So the yellow warning saying there are some cells already classified. So the current 179 cells, probably some of them, you classified them as monocyte or T cell. I don't know which one, but now the later label will overwrite. Now I'm going to call that B cells. So watch those numbers. You're going to see at least one of them will be reduced, but the B cell, you're going to have 179. Okay, I'll type in, I'll call that B cells. So later label will override the previous one. Monocyte, some monocytes, you know, now we'll call it B cell. T cell, it didn't touch, it didn't change, right? Okay, so there's another way. So suppose I see, you know, cluster 4 is kind of, NK cells. I have NKG7 or GLY, GZMK. Those are the cytotoxic cell markers. I want to show you another way to combine the your criteria. Previously, when we select, whatever the criteria you specify here, they will perform AND operation, which means the selected cells, they have to meet all of the criteria specified. But the next one, if I say, I want to select the cells, either they have a high expression of NKG7 or they have a gene LY. I don't want to use an AND. It's kind of too hard. I want to use a OR operation. What are you going to do? So this is how you do it. Let me just color it to make the selection look nicer. Okay, I can see the NKG7 is kind of over here and close to the T cells group, right? So now I'm going to use the NKG7 to select it. The high expression, suppose greater than 7, is 196 cells selected, right? And then once I select it, I will classify, call that, some of them, they probably already classified as T cells, I'm sure. And then they're going to call, I'll just call it NK. Okay, T cell got changed. So NK cells 196. And now I want to use a GLY. So those are 196. They definitely will have high expression of NKG7. And now I want to use, I want to remove this criteria and use a GLY greater than, again, I'll use a 7. So I have 106. So this 106 probably I already called NK cells because of the both the cell have both gene have high expression, but some cells probably is not selected using the using NKG7, right? So now if you will watch this number, NK cells, before we have a 196, you can have a bigger number, but it won't be double. I mean, it's not the sum of those because there's some cells they have, it's double positive. So now I select them, and then I classify selection. I will call them, still call the same name. When you call the same name, Partek will automatically combine them together. So you call that NK. Watch this number. It changed to 205. So a lot of them, they are probably double positive, and some they are single positive. I would highly recommend you, even you close this, it's okay to save your analysis. Now I have a multiple plot, here, suppose I want to take a break. You probably won't be able to finish the classification within one day. You save it. I will save this as my analysis in process. Okay. And then you can log off, flow, or if you want to share this project with your colleague, and you can, choose your colleague name, and later on they can say, hey, go to my data viewer, go to my analysis, and see what do you think. Is where you left off. And if you go to classify, everything is here. So when you in the classify dialog, when you classify even one class, one cell type, you will have in the style configure, you will have this option. It's called new classification. It's just a placeholder, and you can take a look at it. Okay, what my new classification look like? Now I have, I have here. But then you will still see some, NA, some cells is not classified. You can continue on it. Suppose I think this one, I want to call this NA. Now this time I want to change it. I think the whole place, this part is, monocyte, right? So if you can, based on that, you select all the cells, and then you go to classify. I'm going to call that monocyte. So watch this number. Before I have a 323, now I select more on that. You're going to have 353. So got a 20 more cells more classified. Okay, so that's how you continue, keep on going until you say, oh, yeah, I really cannot tell what the cells are. I really don't know. This is my first iteration of my analysis. That's it. So only you finish this iteration of your analysis, you will click on apply classification. So you imagine you have a matrix. Each row is a cell. And now you're going to add a column. For every cell, every barcode, you're going to add the annotation of those 4 cell types. Okay, so you don't want to partially only classify one type of cell and leave the other. Don't do that. So you're going to have one column, have all the annotations of the cell type. So click on apply classification. You give a name, call that my cell type. Maybe you call it a version 1 or version 2 or whatever you want to call that. So once you do that, this information will be saved. If you go to your metadata, will be saved under cell attribute right here. And once the information can be found in the metadata tab, either sample level, if you have multiple samples, you have a treated versus control, all those information of every sample. Of course, all the cells belong to the sample will be the same group. Or the cell level attribute, they're all appear here. That means on your pipeline, every data node can see that information. So you don't have to look for, oh, I want to look at my cell type. Each data node will have that. So if I, now if I, open my UMAP, and you can directly go to the cell type, you don't have to select this. You will have a cell type. So we call that the project level attribute. At the project level, every data node can see that. There's one thing I think is a very useful visualization. I like to show you. I can just build it from here. If I want to build a scatter plot, I want to use the gene expression to build a plot. So we can see how to set up a plot. So suppose I go to the data viewer. I can go to new session. I say, okay, I want to use my gene expression. I want to use normalized count. So I will choose normalized count. I'd drag this data node, say I want to add a plot. What type of plot? I want to do a 2D scatter plot. And then it will ask you what your x-axis, what's your y-axis. So my x-axis, I want to use, suppose, NKG7. And then my y-axis, NKG7. And my y-axis, suppose I will use GNLY. And this is a commonly used plot. You want to look at the gene expression, and we can clearly see those cells, they are double positive. And here is, single positive cells, and this is a 0. Okay, so you can use this kind of plot to make a selection or visualize a certain visualization. All right, so once we have a cell type classified, and all the downstream analysis should be very easy. But the important thing is, the next thing is that I want to perform a differential analysis. Since I only have one sample, I want to compare which genes are showing difference between between the T cell versus B cell, for example. Okay, you can only answer this question. Always look for the data node containing gene expression. Do not perform differential analysis on PCA or UMAP. This does not have a gene expression. I can right click, change the color. If I change to this color, this does not have a gene expression. You might want to start from here. So you always want to, your normalized data node probably is your starting point. You can right click on this and rename it. I will just call that my start point. So you can rename that. It will be easy for you to search for data node. So those are little tricks when you use Partek Flow. So click on that. I will choose differential analysis. We have a different models, and pick your favorite. Depends on your normalization. If you use a Partek default normalization, our default method is ANOVA, because that will fit log normal distribution, or hurdle model is another good option. This model handles zero inflated data. In my normalized count, there's a lot of zeros. That's the nature of a single cell. So either one is a good option, but all the dialogue should be very much the same. You're going to choose, I want to compare cell type. You usually don't want to include all this, those information, because your data is already normalized. You don't have to use this to put as a covariate. So I add the factor of my cell type, and then I want to compare, I'll just choose whatever I want to come to random B cell versus T cell, and then click finish. So this downstream analysis is very much the same. If you do bulk RNA-seq, you're going to do this too. It depends on what method you will choose. So after this is done, there are a couple of plots I would like to show you that is frequently used for single cell data analysis. Double click on this report, you will have a table. Each row is a gene. We have 12,000 genes displayed right here. And for each comparison, you can look at the B cell versus T cell, B cell versus monocyte. You can have multiple comparison, but I only compared those two. And for each comparison, we have the gene name and the p-value, FDR corrected p-value, ratio, and the fold change. And fold change, even the data is in the log space, but the fold change we translate that into linear space. And that's why you see a big fold change, because you look at the mean, if the gene have no expression, it's very, very small, right? It's probably 0. That's the ideal. So the fold change is huge. Some other software, they only have like a 1-digit fold change. That is log fold change. So if you really want that, you can click on the optional column. You can add all those information. And then we call it log ratio. And you can see the log ratio is not that scary, right? If you think so. So of course, on the top gene is B cell marker genes, and you can sort it based on the other way. And this is definitely the T cell marker genes, right? And then for every comparison, this is how invoke volcano plot. So that's the more directly. Of course, you can build a volcano plot from scratch. Basically, your data node has to be a NOVA result, because the x-axis is a fold change. We display in log scale, but because if you display in linear scale, it's going to be you won't be able to see low difference. And the y-axis is p-value. And on each dot is a gene. So upper left and upper right corner are the genes that they have a small p-value and a big fold change. And you can change the gene size, it's the same configuration, right? Depends on what you want to use. Also, if by default we select using this criteria as a p-value, very, 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 very small. You saw that it's e to the minus 100 or something. It's very, very small. But if you use like FDR, go to axis, and you can change. Okay, my y-axis, I can use the FDR. It's still going to be very small to check. So by default, we select this, this part and this part of genes is too many. If you have more than 1,000 genes selected, we won't label that. However, if you just manually select it, I want to select those genes. If you select less than 100 genes, you will see they can clearly labeled out because we changed the location. But if you select more than, I forgot, it's 100 or 300 genes, if you less than 1,000, you won't be able to make them to see it clearly, right? So that's the trick of the volcano plot. If you want to present your result to highlight some genes, usually you only highlight 20 or 30 genes to tell your story. But in terms of the future significant genes, I would recommend you use the left panel. So here you can choose, I want to use FDR. If you have a multiple comparison, you can choose for each comparison. I specify a particular criteria. But here I only have one comparison. Suppose I want to use the FDR 0.001. they have the e to the minus, hundreds. And the fold change, you only have one comparison. You can use 2, or I want to be a little bit bigger, and maybe I can use 3 to select. On the top will give you the feedback. On the 700, maybe it's still too many. Maybe you can change your criteria. So whatever, your criteria is, again, always look at the feedback. So if you're okay with this genes, you click on generate a filter data node. That will give you a data node to only include those 500 genes. Another important plot is for each gene, if you want to see why this gene is on the top of my list, the dot plot is a very nice visualization you might want to use. So when you have, this is a 2D scatter plot, each dot is a sample, but the x-axis is a categorical variable, y-axis is a numerical variable. When you have this type of plot, you can also, in the style, add a violin on it, overlay the violin and make it the color, or you can turn off the turn off the point, display. So this one type of plot actually can be multiple plots. Depends on which one will help you to tell the story. And another nice thing is if you want to display multiple genes, so suppose I would say less than 10 genes, you can easily duplicate this plot and double click on the y-axis and search for your gene. Suppose I want to look at a CD14. And then you can, have multiple plot here and try to display, double click on the y-axis to change different genes you want to display. So when you export the image on the left side, they will export all the image together. And when you use control to select multiple image in the style, you can configure them and all at once, make them bigger or smaller of the point. One last thing I want to talk about is if you have a lot of genes, so here I have 500 genes, a better way to help you tell the story is do a hierarchical clustering or heat map. So on this gene list, and you will choose the heat map. The heat map is a color presentation of the matrix. Okay, so the matrix, we're looking at each cell for each gene, but we only look at 500 genes for all the cells. And you can choose how you want to order them. If you say, I want to use a cluster to order them, you don't have a control. It's based on statistics. You will have a dendrogram. Or if the cell, if you say, I don't want to order the cells, I don't want to cluster the cells, I want to order them manually, then you can choose, okay, for the cell, I want to order them based on the cell type. And you can change, how you want to order them. Also, you can filter it because this 500 genes is the only difference between B cell versus T cells. If you include the other cells, your picture won't look good. So it's better that you only include, because you're going to tell the story that those cells can differentiate between B cell versus T cells. So I don't want to include all the other cells to make it look too messy. So now this is the hierarchical clustering at cell level. If I open that, and here is the barcode, that's the cell level, and this is the 500 genes. You can scroll your mouse wheel to zoom in. And some key configuration is, suppose on the axis, you can turn off the barcode. Instead, you can add annotation. All the configuration is the same as all the other. If you have bulk RNA, it's going to be the same. You can add the cell type. And now you clearly see the genes cluster. Also for dendrogram, we can, the block size, you can change to, for the dendrogram, I want to color my, based on 2 cluster, you can clearly see this group of the genes have a high expression in B cell, and this group is the other way around. The heat map coloring over here, you can change the scale, depends on what do you think, how it look like, and you can add more colors Depends on what you think is, you can use more than, any color, as any number of color as you want. Suppose I want to make an outlier as pink or purple, whatever. So you can add more colors to look at those.